Students, from genetics, we are into another topic, that is mutagenesis. If you see here, mutagenesis. What is mutagenesis? Mutagenesis is a process by which the genetic information of an organism is changed, resulting in a mutation. That is mutagenesis. It is a process by which the genetic information of an organism is changed, resulting in a mutation, that is mutagenesis. So it may occur spontaneously in nature or as a result of exposure to mutagens. It can also be achieved experimentally using laboratory procedures. In nature, mutagenesis can lead to cancer and various heritable diseases, but it is also a driving force of evolution. So, if you see here, there are different types of mutagenesis. There is here you see directed mutagenesis, site directed mutagenesis, PCR mutagenesis, insertional mutagenesis, signature tagged mutagenesis, transposition mutagenesis, sequence saturation mutagenesis. If you see here, directed mutagenesis, directed mutagenesis, also known as directed mutation, was a hypothesis proposing that organisms can respond to environmental stress by orthogenetically directing mutations to certain genes or areas of a genome. So, if you see here, it is nothing but a directed mutations. Uh, is that it in this what the organism can respond to any kind of environmental stresses which results in these mutations. There are different types of directed mutagenesis, that is oligonucleotide directed mutagenesis. If you see here with M13 DNA, oligonucleotide directed mutagenesis with plasmid DNA, PCR amplified oligonucleotide directed mutagenesis. So what are these advantages? If you see, mutation rate is high here, all mutations can be induced, systemic and detailed investigation of target mutation can be made. So, if you see here, site directed mutagenesis, if you see, is a molecular biology method that is used to make specific DNA and intentional changes to a DNA sequence of a gene and any gene products. So, especially this is used when in a biological method or any biotechnology method, it is used to especially to make a specific intentional changes where to the DNA sequence of a gene and or any kind of a gene products. So if you see, it is used mainly for investigating the structure and biological activity of DNA, RNA and protein molecules and for protein engineering. Next one, this is a diagram which illustrates the mutagenesis, site directed mutagenesis. Here how it has been targeted and how it has been changed. Like in here it is used in the propagation of E. coli. This is an example. Next, if you see here, there are some kind of mutagenesis, that is signature tagged mutagenesis. STM is a genetic technique used to study gene function. So, to study the gene function, this STM, that is signature tagged mutagenesis, has been used. So, recent advances in genome sequencing have allowed us to catalog a large variety of organisms. If you see here, the original and most common use of this STM is to discover which genes in a pathogen are involved in virulence in its host and to aid the development of new medical therapies or drugs. In short, we can say this STM, that is a signature tagged mutagenesis, is used to find out which genes in a particular pathogen is involved in a virulence in its host, and it also helps in development of new medical drugs or discovering the drugs or preparing any medical therapies or anything like that. So this is a diagram which indicates here how it has been prepared. Next, transposon mutagenesis. Transposon mutagenesis is also called as transposition mutagenesis. This is also a biological process that allows genes to be transferred to a host organism chromosome, interrupting or modifying the function of an extant gene on the chromosome and causing mutation. Here, this is also one kind of a mutagen, and it is a biological process in which allows the genes to be transferred to a host organism's chromosome, interrupting or modifying the function of an gene on the chromosome and causing the mutation. So transposition mutation, here you can see insertion of transposition within a gene leads to mutation. Transposition with antibiotic resistant marker can be used for selection process. If you can see how it has been used and disrupted here, the diagram. And next one, if you see the applications of this mutagenesis, site directed mutagenesis is used to study changes in, if you see in the protein activity that occurs as a result of DNA manipulation. It is used to access activity of proteins containing known amino acid substitutions, and it is also used to select, screen or screen for mutations at DNA, RNA, or protein level, which can be manipulated further for enhanced biological activity. And it is used to change specific amino acid in an enzyme, and also it can be modified, it can be used in the modifying enzyme molecule. And if you see here, the site directed mutagenesis helps scientists to study the mechanism of action behind biological reactions, etc. So applications, so here if you see where it has been used, applications, if you see, recombinant DNA technology and use of synthetic DNA molecules make it possible for molecular biology scientists to induce specific mutations in specific genes. There it can be applied. In vitro mutagenesis is used for purposefully to change the genetic information, and also it is used in enha- like analysis of some gene expression, which helps to elucidate or isolate any functional effect of the mutation. And there are various types of mutagenesis, as I told you, site directed mutagenesis, random mutagenesis, etc. So if you see here, screening and selection. What is a screening and selection? So, genetic screen or mutagenesis screen. So, in mutagenesis only, this is one type, that is the genetic screen or mutagenesis screen is an experimental technique used to identify and select for individuals who possess a phenotype of interest in a mutagenized population. Here, if you see here, especially it is used to select for individuals who possess a phenotype of interest in a mutagenized population. That means genetic screens can provide important information of gene function as well as the molecular events that underlie a biological process. So these are the like screening technique can be used in this mutagenesis. So if you see here, these are the basic screening, that is forward genetic screen. The screen, especially this type of a genetic screen, is to know the interest or phenotype interest of a mutagenesis population. This is the enhancer scheme begins with a mutant individual that has affected process of interest with a known gene mutation. That means any mutation has been affected or interrupted with a known gene or disrupted with a known gene. Okay, probably to isolate or to identify the interacting genes, what is happening inside it. Next, temperature sensitive. A temperature sensitive screen involves performing temperature shift to enhance a mutant population. Here, an advantage, if you see here, a null mutation such a gene may be lethal to the embryo, or such mutants will be missed in basic screen. That means here, if you see here, what is this temperature screen sensitive screen? Here, if you see a screen for temperature sensitive in fruit flies, for example, might involve raising the temperature in the cage until some flies faint and then opening a portal to let the others escape. So this is one kind of a mutation can be caused. Suppressor screen is used to identify Suppressor mutations, especially suppressor mutations, can be described as a second mutation at a site of a chromosome distant from the mutation which has been in a suppressive stage. So, suppressor mutations are extremely useful to define the functions of biochemical pathways within a cell and the relationship between different biochemical pathways. So, if you see here, screening selection, mainly three types of screening selection techniques, that is visual, mechanical, physical, and some other methods can be done. Visual screening can be done is most effective and efficient method for identifying mutant phenotypes, and also visual selection often is prime basis, especially used for selecting disease resistance, earliness, plant height, color changes, any kind of ions shattering, or any plants which is can be adapt to soil, climate, or growing period, etc. Mechanical or physical, if you see, very efficient for seed size, especially used in agriculture, shape, weight, density, etc. Using appropriate sieving machinery. Example, if you see here, they can be checked with chromatographic or electrophorese technique may be used to select isolate protein variants. Methods for validation of mutants, that is, it can be used genome wide chips, different screening, microarray, PCR screening, tilling, and echo tilling. So here if you see, DNA chip technology utilizes microscopic arrays of molecules immobilized on solid surface for biochemical analysis. So if you see tilling or echo tilling is a general reverse genetic technique that combines chemical mutagenesis with PCR based screening to identify point mutations in regions of a specific interest. Okay, tilling is a very powerful technology that employed to detect which organisms in a particular population carry nucleotide mutation specific genes. Echo tilling is similar to tilling except that it is objective to is to identify natural genetic variations. If anyone wants to identify natural genetic variations, this echo tilling can be used and opposed to induce mutations. These are the different methods for generating mutant varieties. If you see in mutagenesis, forward genetics, reverse genetics, here forward genetics is through chemicals and radiations. Reverse genetics is insertional, agrobacterium mediated transformation, virus induced gene slicing, RNA mediated interface, tilling process, etc. So, what are the advantages? Induced mutagenesis is used for the induction of cytoplasmic male sterility, and also is mainly used. Ethidium bromide has been used for induction of cytoplasmic male sterility in pearl millet and barley, especially millets and barley. Mutation breeding is a cheap and rapid method, developing a new variety as compared to the backcross pedigree and bulk method. So mutations breeding is more effective and simple and quick and best way with when a new character is to be induced in vegetatively propagated crops. What are the disadvantages? If you see, most of the mutations are deleterious and undesirable. Identification of mutations which are more useful to plant breeder is usually very difficult. Mutation breeding has a limited scope for genetic improvement of quantitative and polygenic characters, and also this isolation will be more difficult for them for desired mutations. So these are the future things. So with this we finish the mutations. Hope everyone understood this. So mutations is a very vast topic. What I have gave the all the probable terms, everything for y'all in this. Thank you.